everyone, and we are going to start with the next part of this chapter, that is sexual reproduction in flowering plants. In the previous lecture we studied about the structure of anther, the internal structure of anther, even the wall, that is the 4 layers of anther wall. Now inside this anther takes place the process of microspore formation, and that process is known as microsporogenesis. So we will talk about that process. Once the microspores are formed, the same microspores will change into the male gametophyte, that is the pollen grain, and we will see the structure of pollen also. Pollen grains are also consumed in the form of tablets or syrups. What is the reason behind it? That also we will discuss. Okay, so let us start with the basic information. This is about me, and I guess now you are all aware of the name, that is the teacher who is taking your class. This is about the Telegram channels and the steps. See, we have started taking the quizzes and the doubt solving sessions on Unacademy learning app, and most of you have attended those also. But in case somebody has not attended those doubt solving sessions and quizzes, they need this app, Unacademy learning app. So install this app, very simple steps. And in case if you decide to go for any subscription plan, don't forget to use my code, which is going to give you 10% discount, and then apply this code. You will have that particular subscription plan. Now, there are a couple of programs which have already begun, and there are some which are going to be starting very soon. So this is one, Pehel, and it has already begun. It started on 28th, but if you want, you can still join this. These are the teachers who are taking classes there. Another program or a batch which has already begun from 28th of December is called Super Ranker Batch, and all these are targeting NEET 2021. The teachers who are taking classes there, these are the names, and this is some information about them. The third one, which is going to start from January 3rd, is called Umeed Batch, and these are the faculty members who would be taking classes for you there. This is our timetable for Conquer NEET 2021, right? So I gave you the timetable till 2nd, and after this the new timetable I'll upload in the next session, so that we know what exactly is going to come up. But don't forget to install the app, because after every chapter or in the middle of the chapter we take doubt solving sessions and some part quizzes. So if you want to participate in those quizzes, you will have to install the app. This slide is from our previous lecture. This is the structure of anther that we studied, and these are the 4 layers of our anther wall: epidermis or epithelial layer, also endothelium, middle layer, and tapetum. Tapetum is the nutritive layer. It provides nourishment to the developing microspore, and here inside there are these archisporial cells. They are also known as archegonial cells, diploid cells. So these archegonial cells are diploid, and they are the ones which actually will give rise to the microspore. Now, microspore mother cells will be formed from these same archegonial cells, and they give rise to microspore. So this process will be termed as microsporogenesis. So what exactly we mean when we say microsporogenesis? It is microspore mother cell to microspore formation. This process is called microsporogenesis. Reproduction. Now to understand this, what we will do is, we will draw a diagram which is going to help us understand what happens as the anther matures. And again, let us make this diagram little bigger so that all the stages can be drawn inside this. Normally in the diagram itself we should be able to show all the changes. Just so that our diagram looks little neat. This dotted line helps us make a symmetry or symmetric diagram. Okay. This is our anther and we know this is one lobe. So bilobed or dithecous anther is what we are talking about. And you remember we made a sac-like structure here. So what we will do is, now we will make all the changes inside this one diagram. So this is stage number 1 which we are drawing. 1. Then the next stage we will make here. 2. The next stage we will make here. This is going to be our 3rd stage. And the 4th one we will make here. So in one diagram we can show all the stages. Now let us come to the first stage. In the first stage, if you remember, we packed this sac tightly with cells. The sac was called Microsporogenesis and the cells are archegonial or archisporial cells. And let us come to this. The sac is called the Microsporogenesis. This first stage is seen when the anther is young. Now as anther matures, the entire structure is going to grow in size. So when the entire structure grows, even the sac-like structure becomes bigger. So the sac becomes bigger, here also it becomes bigger, and here it becomes bigger. So we will see all stages, and now we will start calling it pollen sac. So in young anther, those sacs are known as Microsporogenesis, but as the anther starts to mature, we start calling those sac-like structures as pollen sac. Now what happens? As the sac, the compartment or the area becomes bigger, that means the cells which were tightly packed, now they become little loosely packed. Spaces appear between those cells because now the sac has become bigger. These archisporial cells are diploid. Now when these cells, they get loosened up a little bit, we start calling them microspore mother cells. Microspore mother cells. And these microspore mother cells, they are also diploid. So if we make this microspore mother cell, which is diploid, it undergoes meiosis because it has to produce haploid structures. So when meiosis takes place, one parent cell gives rise to 4 daughter cells. So here we will see One cell, second, third, and fourth is on the other side. And these cells are haploid, but all four of them remain together. So this is called a microspore tetrad. If I have to just show you that this is one cell, the second cell, the third cell, and the fourth cell is behind it. These four cells, they are glued together. They are together. And what is that thing which is helping them to remain together? Is callus. So four cells are formed, microspore, four microspores are formed, but they remain together. They are stuck together with the help of a material. That material is called callus. Now this microspore tetrad, it is made up of four haploid microspores. Do you want that these four cells should get separated? Because each microspore is going to change into a pollen grain. So if these four cells have to separate, what do we need? This glue material or gluing material should be dissolved. So callus is digested by enzyme callase. Do you remember which was that structure that secreted callase? It is secreted by tapetum. So tapetum is that layer which secretes this callase. So now, because of this callase, what happens to these microspores? All four microspores, they get separated. So these are the four microspores. And because they are formed as a result of meiotic division, they are haploid. They have half the number of chromosomes as that of the parent cell. So if microspore mother cell has 20 chromosomes, the microspores will have half the number, that is 10. Because inside these microspores, which is going to change into pollen, male gamete will be formed, and the gametes have to be haploid here. Now, let us talk about this microspore tetrad. There are various shapes or arrangements in which we can find these microspore tetrads. Okay, one which we have drawn already, where you are able to see 3 cells and the 4th cell is at the back of it. The second arrangement is where you are able to see all 4 cells. Okay, then there is one arrangement where we are able to see all the 4 cells, but they are like 1, 2, 3 and 4. This is one pattern in which they are arranged. One more arrangement is there where we see these 2 cells like this. One cell here and one cell here. This arrangement is also possible. One more arrangement is there where you see 1, 2, third is here, and the 4th large cell. So these are the possible arrangements of 4 microspores in a tetrad. Okay, and now if we have to give them the name, this is called tetrahedral, and this is the most common. And that is why we made this, because we normally draw the structure which is most commonly seen. The second one, this one is known as isobilateral. The third one is very simple. It is called the linear one. Let us not forget to draw the nuclei, because these are all haploid cells. So tetrahedral is the most common one, which is seen in most of the angiosperms. And why are we using the term angiosperms here? Angiosperms are the flowering plants, and our chapter is sexual reproduction in flowering plants. So basically we are talking about angiosperms. So in most of the angiosperms, the microspore tetrad shape which is seen is tetrahedral. Then some have isobilateral also, some have linear also. This is known as F-shaped, and the last one is known as decussate. These are not that common. So isobilateral, linear, F-shaped, decussate, not very common, maybe 2%, 4% like that. But majority of microspores are, or tetrads, are in the form of tetrahedral arrangement. Now, what would you see in the third stage? Here what has taken place is meiosis in the microspore mother cell, and now pollen sac has become even bigger, and here we are seeing those microspore tetrads. Okay, so these are microspore tetrads. And these 4 microspores are held together because of callase, and the enzyme, callose, and the enzyme callase dissolves this callose so that all 4 microspores are set free. So microspore formation is over, and that is the process of microsporogenesis. Now here we will take one more example. When we talk about microspore formation, this is like the most common one. But in Calotropis, the pollen sac, they form such balloon-like structures, and the pollen grains are packed inside them. So the pollen grains will be here. Such type of a structure is called pollinia. This structure is known as pollinia or pollinia. It is seen in Calotropis. Now, what is the significance of the pollen sac to remain in this balloon-like structure? This we will take up again in adaptations for cross-pollination and self-pollination. There is a physical barrier. Say for example, if this is the stigma and a pollinium comes and falls on this like this, and pollen grain is inside. So pollen and pistil will not come in contact with each other. So there is a physical barrier between the pollen grains and the pistil, so that cross-pollination can take place. These pollinia will move through wind with the help of air. They will move from one place to another, from the plant where they are released to some other plant of the same species. And by that time their membrane becomes delicate, thin, dry, so that it can break off easily. So the purpose is that this particular pollen should travel great distance and go to some other plant of the same species so that cross-pollination can take place. If polonium is formed and it falls on the stigma of the same plant, because the wall of that polonium is still fresh, hard, though that pollen and pistil will not come in contact with each other. So this comes under an adaptation. We'll talk about this again when we come to pollination and adaptation part. So remember, pollinia, these are balloon-like structures which can be carried by wind, or they may stick to the body of insects also, and this is seen in case of Calotropis. So now in first stage, in young anther, the cells diploid itself were compactly arranged. We call them archegonial cells. Then as the sac becomes bigger as the anther matures or starts to mature, those cells, they become spaced out. There is some space between the cells. We start calling them microspore mother cells, which are diploid. Microspore mother cells undergo meiotic division and results in formation of 4 haploid microspores which remain together in the form of a tetrad. Now why do they remain together? Because there is a glue-like material called callose. Now the callose has to be dissolved. So the enzyme, and remember the enzyme is coming from these cells. These cells are the tapetal cells. So tapetum or tapetal cells, they secrete these enzymes. They do many jobs, and we have done this. We have written a list of things which are done by tapetum. It is a nutritive tissue. It secretes sporopollenin, which we will talk about in some time, enzyme, callase, and also help in pollen pistil identification. Now there is one more thing which we can add here about tapetum. These cells are pyramid shaped. We talked about this. We made the shape like that. These are those cells, and they are either binucleate or multinucleated cells. So their number of nuclei inside them is more, or sometimes they have one nucleus but the genetic content is more, because they have to synthesize more and more material. So this is about tapetum. Alright, so now this is the part which is called Microsporogenesis. Clear this much part? Now, okay. Formation of male gametophyte. What is a male gametophyte? Male gametophyte is the pollen grain. Male gametophyte is the part of the plant which produces the male gamete. That is known as the male gametophyte. So now let us take this same thing, that is our microspore. So these microspores are now separate. This was our microspore tetrad, and these microspores are now separated. Microspore tetrad, and these are microspores. Now each microspore is going to change into a pollen grain. So now what happens is, this microspore becomes bigger and its nucleus divides by mitosis. mitotic division of nucleus, only karyokinesis. So now there are two nuclei, and because it is a mitotic division, here the nucleus was haploid and here also both the nuclei are haploid. Both nuclei are haploid. Now, there will be cytokinesis, cytoplasmic division will take place. But cytokinesis takes place in a slightly different manner. One nucleus gets surrounded by the membrane, which is made up of callose, and it is on one side. Normally it is a spindle shaped structure. That means now there are two cells, one larger cell and one smaller cell, which is actually inside the larger cell. So after some time we will find that this larger cell has its nucleus and the smaller cell, which is spindle shaped, is now actually floating in the cytoplasm of the larger cell. Okay, so this bigger cell is called the vegetative cell, and this smaller cell is called the generative cell. Now while this is happening, the larger cell secretes, cytoplasm of the larger cell secretes a layer around itself. This layer is called intine. And now outside this intine, tapetum secretes sporopollenin, and that sporopollenin deposits here. So now what will happen is, this sporopollenin, it gets deposited here outside this. This layer is exine, and the structure which is formed is actually the pollen grain, and inside the pollen grain will be the male gamete formation process. Okay, so this pollen grain which is formed is a two-celled stage. There are two cells, one vegetative cell and one generative cell. In most of the angiosperms, about 60% or more, the pollen grains are released in two-celled stage. Very, very important. In most of the angiosperms, pollen grains are released in two-cell stage. The male gamete is still not formed. So this two-celled pollen grain is called the immature male gametophyte. Why immature? Because the male gamete is still not formed. Clear so far? What exactly has happened? The microspore tetrad now separates, and there are 4 microspores now. What was that reason which helped in separation? Was an enzyme callase secreted by tapetum. Each microspore undergoes karyokinesis, mitotic karyokinesis, and then cytokinesis, resulting in formation of two cells, one larger, one smaller cell. Okay? Then a layer is secreted by the cytoplasm, which is called intine. Then there is one more layer which is actually deposition of sporopollen. So this structure is now called pollen grain, but it is immature because here male gamete is still not formed. So let us now draw the structure of pollen grain, detailed structure of pollen grain, and then we will come to the mature pollen grain. We are making it slightly bigger. This is the bigger cell, the vegetative cell, its nucleus, and here is the other cell, which is the smaller cell, and it is spindle shaped. Okay, let us write down about these two. This is vegetative cell. Vegetative because it does not participate in male gamete formation. Vegetative is not helping in sexual reproduction. And it is haploid because it is formed as a result of meiotic division. Generative cell is the one which will give rise to the male gamete, and this is also haploid. So this will give rise to two male gametes. So what is the function of this vegetative cell? The vegetative cell performs two functions. One, it has to provide nourishment to the generative cell and then the male gamete. Provides nourishment to generative cell and then the male gametes. And it is the same cell which will later on give rise to pollen tube. Will give rise to pollen tube. And that is why sometimes vegetative cell is also known as pollen tube cell because of its function, that it is going to give rise to the pollen tube. Now, the generative cell, its wall is made up of callus. Its cytoplasm is clear without any reserve food. So where is the reserve food? The complete reserve food is with the vegetative cell, and that is why it is able to provide nourishment to the inner cell, that is the generative cell. Now, outside this cell, the larger cell, the cytoplasm itself secretes a layer. And this is made up of cellulose. So cellulose is secreted by cytoplasm of the vegetative cell. And this layer is called intine, secreted by cytoplasm of the vegetative cell, and it is made up of cellulose. Okay, and now tapetum secretes sporopollen, and this sporopollen slowly gets deposited outside the intine. Now when sporopollenin gets deposited, because it is getting deposited from outside, the outer surface is not smooth. It is rough, irregular, and it is a thick layer, and this layer is called the exine. And you can see that there are certain places where this exine is not deposited. So this layer is called exine. What is it made up of? It is made up of sporopollenin, and this sporopollenin is a substance which is resistant to acids, temperature. So we normally write it as most resistant biological substance known. So it doesn't get affected by acids, it doesn't get affected by temperature, high temperature, low temperature, nothing affects it. And that is why we call it most resistant biological substance. And if an animal, say, eats this pollen grain, so nothing is going to happen when it passes through our alimentary canal, because in our alimentary canal there is alkaline pH also, acidic pH also, there are enzymes. So it is resistant to, sporopollenin is resistant to acids, bases. It is resistant to temperature, and it is also resistant to enzymes. Now what exactly this sporopollenin made up of is not very clearly understood, but some scientists believe that it is a combination of lipoprotein, it is a combination of polysaccharide and lipids. So there are multiple things, but we don't exactly know what it is made up of. Now if it is so resistant, then how come the pollen tube emerge from it, from the pollen grain? Because pollen tube is a tube-like structure which has to come out, so there has to be some weak area from where that pollen tube can come out, and this is the weak area, and this is called the germ pore. Sometimes it is circular opening, so we call it pore. Sometimes it is a little longer opening, so we can call it germ furrow. Now what exactly is this germ pore? Place where exine is absent. There is no exine. Intine is there. But because it is a weak spot, the pollen tube can emerge from here. And these are very tiny openings. It's not like a very big opening, but this is a two-celled pollen grain. Two cells, vegetative, larger, and generative, smaller cell. Intine, the inner layer which is secreted by the cell itself, and exine which is secreted by tapetum. Made up of sporopollenin, and it is secreted by tapetum. And it is immature because there is no male gamete. So it is the pollen grain, and in most of the angiosperms, the pollen grain is released in this stage itself. Okay, let us go back to this stage. And now, when this matures, this is what we have to remember, that at this stage, pollen is released. And now this pollen from the anther will go to the stigma. That process is called pollination, that we will take up next. But in 60% or more angiosperms, it is released in this. What about the other 40%? So in the remaining 40%, what happens is, this is the vegetative cell, its nucleus, and the generative cell undergoes one more mitotic division. So here we have to write in about 40% angiosperms, the generative cell undergoes one mitotic division to form, and it is mitotic division. Generative cell was haploid, vegetative cell was haploid. So if haploid generative cell is undergoing mitotic division, so the two daughter cells will be haploid to form two haploid male gametes. So these two are the haploid male gametes. Now, how many cells do we see? We see three cells. One is the larger vegetative cell and two male gametes, and the intine is also here, and outside the intine, exine would also be there. So this would be a three-celled pollen grain, or we can call it a mature pollen grain or a mature male gametophyte. So this one is mature male gametophyte. What happens in the majority of them, that is 60% of the angiosperms? In 60% of angiosperms, pollen grain germinates when it reaches the stigma, and that is when the generative cell divides. So when it reaches on the stigma and the generative cell divides, that is when we call it a mature male gametophyte. So in majority of angiosperm, the pollen grain is released in the form of an immature male gametophyte. But when it reaches the stigma, the generative cell divides to form two male gametes. That is when we say that it is a mature male gametophyte or germinating pollen grain. But in some angiosperms, it's normally 40% or less, the pollen grain is released in three-celled stage. Okay? In about 40% angiosperms, pollen grains are released in three-celled stage. In majority, they are released in two-celled stage. So now we know what is the difference between a mature and an immature gametophyte. We also know how this pollen grain is formed, and when we say two-celled stage or three-celled stage, what is the difference? All right. So let us come back to this original stage. Now, can we make the pollen grains in the final stage? So now here we actually see these pollen grains. That means the anther is mature and all 4 pollen sacs are filled with pollen grains. Okay, now let us go back to those 4 layers of the anther wall. If you remember, we made the outermost as the epidermis or epithelial layer. We'll make it here again, just so that we understand how this pollen which are formed can be now released. So this is the epithelium. Below the epithelium are present some layers and 2-3 layers of cells. This is called the endothecium. And the other name of this was fibrous layer. And inner to it were the cells of the middle layer. Innermost layer is tapetum. The cells are pyramid shaped, right? So let us make few pyramid shaped cells here. This is tapetum or tapetal cells. Okay, now when the anther is young, all 4 layers are as it is: the epidermis, the endothecium, the middle layer and the tapetum. Now as anther matures, the endothecial layer, it turns into fibers or it becomes fibrous. So if this layer becomes fibrous, and see where it becomes fibrous almost everywhere except some places like this place and this place. Now when the cells become fibrous, they become tight, dry, and when they become dry, they sort of shrink. So when it shrinks, the pollen will be released. And how will the pollen released? It is going to rupture from here. Let me make this. So here the pollen ruptures. Sorry, the anther ruptures. Anther ruptures. When the anther ruptures, this pollen sac will also rupture and the pollen grains will be released. So these layers, the outer layers like epidermis, endothecium, middle layer, they are the ones which will help in releasing of the pollen grains. And how would this happen? The endothecium in this part, that is in the circular part, will become fibrous. Here also it will become fibrous, and when it becomes fibrous, it becomes tight and it starts to shrink. When it shrinks, if this is the structure and on these places it is become fibrous, so it shrinks like this, and there is a crack or an opening here. So here will be one opening and here will be one opening and then the pollen sac will rupture because of that shrinking or rupturing of the layers, and then the pollen grains will be released. So here we have shown the ruptured pollen sac also. And when the pollen sac ruptures, pollen grains are released. Now in majority of angiosperms, pollen grains are released in 2-celled stage. But in some, like less percentage of angiosperms, the pollen grains are released in 3-celled stage also. That percentage we have to remember. So here we wrote two important things, that in majority of them in 2-cell stage, and in some, like about 40%, they are released in 3-celled stage. So we now know what exactly is microsporogenesis. So let us go back to this part. This was the one that we talked about in the previous lecture. So microspore mother cell to microspore. This process is called microsporogenesis. Then microspore to pollen grain is known as the male gametophyte formation stage. And then inside the male gametophyte, the male gamete will develop. This is microsporogenesis, and in one diagram we have shown all the stages. In the first stage, that is the young anther stage. This is the young anther stage. We are showing all 4 layers, and inside the sac, which is known as the microsporangium, there are tightly packed cells, archegonial cells. Then as the anther starts to mature, the sac becomes bigger. We start calling it pollen sac. The cells are also loosened up, and now each cell is known as microspore mother cell. What happens further? The anther matures further, and each microspore mother cell undergoes one meiotic division. So one meiotic division will result in formation of 4 microspores. So microspores are going to be haploid because they are formed as a result of meiotic division. Okay? Initially, these microspores, they remain together because of a glue-like material which is called callase. This callase has to be dissolved so that microspores can get separated. This takes place with the help of an enzyme called callase. The material was callose. The enzyme is callase. So the callase, which is secreted by tapetum, dissolves this callose, and now the microspores are separate. The microspores, when they are together, that is called tetrad, microspore tetrad. And there are various types of microspore tetrads which are reported. The most common is tetrahedral, and in Calotropis there is a special structure which is called pollinium. Microspore tetrads separate. Each microspore gives rise to a pollen grain. So microspores are changing into pollen grain. This is the formation of male gametophyte. The pollen grain has 2 cells, one larger vegetative, one smaller generative. Maximum nourishment is in the vegetative cell because it has to provide that nourishment, and the generative cell is smaller with clear cytoplasm because its job is to give rise to the male gamete. Okay? Structure of pollen grain is also clear. Now let us come to this last part, which is the pollen tablets. Nowadays there is a trend of supplements, nutrient supplements. So you need something which provides you all those supplements, minerals and everything in a concentrate form. Now if you remember the structure of pollen grain, we said there are 2 cells, right? This was the vegetative cell. And vegetative cell's job was to provide nutrition or nourishment to the developing male gametes and the generative cell. That means it has all that nourishment. So if there is some nutrition inside this, why can't we take this? So nowadays pollens are crushed, their nutrients are extracted and we make tablets out of it. We can make some viscous liquid also, which is called the syrup or syrup, and these are used as nutrient supplements. These are getting very common these days, but in developed countries these are very commonly available. Slowly and gradually this is sort of spreading everywhere. So like we used to take tonics, if you are not getting all the minerals and vitamins and everything, multivitamin tablets or tonics are given to you. So this is another way of giving you the nutrient supplements. All right, so the processes that we have talked of are many processes. There are multiple steps, and I will tell you one more thing here, that whenever you do or revise this, draw the diagram and around the diagram write everything, so that when you see this complete flow chart or the line diagram or whatever you call it, every information is right there in front of us. Like we have written microspore tetrads, we have drawn them, and here itself we have written which is the most common one. So you don't have to read the text at the time of revision. Same is the case with gametophyte formation. Here itself what is happening, first division, second division, and so on. Now if I ask you a question here, from microspore mother cell to formation of male gamete, how many divisions are required? From microspore mother cell, which is diploid, to male gamete, which is haploid, how many divisions are required? Our answer is going to be 1 meiotic division and 2 mitotic divisions, right? Because microspore mother cell, meiosis, we got the microspore tetrad or the microspore haploid. In microspore, mitotic division, so this is 1 meiosis, 1 mitotic division, which results in formation of 1 larger cell and 1 smaller cell. Then the generative cell undergoes second mitotic division to give rise to 2 male gametes, right? So to form these male gametes, there is 1 meiotic division required and 2 mitotic divisions. And if we write from microspore mother cell to male gametophyte or pollen grain, when we simply write pollen grain or male gametophyte, we have to take the majority of, that is 60% of angiosperms. So here our answer is going to be 1 meiotic and 1 mitotic division. So questions are asked in this manner also, and that is why we have to understand the entire process in detail, and that is when we are able to answer such type of questions. Okay, so this is the timetable, which most of things we have completed. Now in the next lecture we'll talk about the female reproductive part. And this is again a last thing, a reminder that if you want to take a subscription, use my referral code, that is Neela, so that you are able to get 10% discount. Alright, so don't forget to read NCERT, whatever we have studied so far, and if you have not downloaded the app, download it so that whenever we come to this doubt solving session and the quiz on Unacademy Learning app, you should be all ready. And while reading, Write down the doubts in that special doubt copy so that whenever we discuss, you have all your doubts cleared. Okay, so I will see you in the next class. Till that time, take care and wish you all a very happy, successful, safe, prosperous new year. Bye.